Hello and welcome back. In this mini lesson, we are going to cover ossifications, the next section in the PowerPoint and the lecture notes that you have from Blackboard. So there are two types of ossification processes we're going to talk about. The first one's called endochondral, which means inside cartilage, basically. And the other one is called intramembranous, which means inside membrane. So it's basically describing the origins of where the bones actually start from. But before we jump into the endochondral, just to define some terms and the differences between the two, we have calcification means, let me look on my notes here, the deposition of calcium salts into a tissue. So just putting calcium into a tissue. So you can have calcification of lots of different things. Unfortunately, you can have calcification of arteries, you can have calcification of kidneys and testes, but that doesn't necessarily mean they're going to end up being a bone, an actual organ of a bone. But the term ossification is basically calcification with the end result of being a bone. So ossification is calcification ultimately to become a bone. Okay, replacing other tissues with bones. So what's cool about the skeletal system, the skeleton itself, is it starts off as one type of tissue and completely turns into a different type of tissue. We don't really see that in many of the other organ systems. So when the heart is being formed in utero, it starts, I got my cat friend here. Say hi. It starts as cardiac muscle and will become cardiac muscle. Your skeletal muscles are skeletal muscles, but your bone either starts off as cartilage and then turns into bone, or your skeletal system starts off as cartilage and turns into bone, or starts off as another type of connective tissue and turns into bone. So this is unique development of your bones, which is kind of cool. So the first type we're going to take a look at is called endochondral ossification. So the name implies, there we go. Okay, endo meaning inside, chondral, remember chondra is a root that means cartilage. So endochondral ossification, we start off with cartilage and eventually we're going to turn it into bone. So the cartilage that we are going to be dealing with is hyaline cartilage. So you learned our three types of cartilage back in chapter 4, remember? So hyaline, elastic, and fibrocartilage. So hyaline is what we start off with in our fetal skeleton. So when you are developing in utero as a fetus, your skeleton is made up of cartilage to most of the bones. I should say most of the long bones are going to be cartilage. Intramembranous covers some of the other ones. So let's say you have your femur, right? In utero, this femur is femur shaped, but it's made up of hyaline cartilage. And then during this process of endochondral ossification, the cartilage model gets replaced and turned into the bone model. And that's going to be your femur bone for the rest of your life. Okay. So there are 6 steps in this endochondral ossification. We're just going to walk through them and you can go back and review the video if you need to. So remember, because it's cartilage, it's going to have all of the same characteristics of hyaline cartilage. It's going to have chondrocytes living in lacuna, surrounded by this gel-like matrix, covered by perichondrium, that connective tissue sheath. So the first step is the chondrocytes towards the middle of this bone. Well, I think it's the humerus. They're going to eventually turn it into the humerus. Oops. There we go. Got my collection of bones. So this bone that they're going to be tracking in this diagram will eventually become the humerus. So we'll say in the middle part of the cartilage model, so pretend this is the cartilage model, the chondrocytes in the middle, in the diaphysis, start to enlarge, their lacuna enlarge, and eventually they die. But that space, the matrix, which is cartilage matrix, calcifies before they die. So it calcifies, it gets solid, like a little spicule almost. And then the cells inside die, so you're left with these big open pockets with calcified matrix around it. And then we have blood supply growing to that surface, right? So now we're in step 2. So around that perichondrium, we have blood supply, and with the blood supply come some bone cells, some stem cells that kind of convert into osteoblasts, right? So wherever we have osteoblasts, so cells of the perichondrium, convert into osteoblasts, and where we have blasts, we're going to lay down bone. So it starts laying down actual bone tissue, not cartilage, around the outside edge of the diaphysis. And sometimes this is called the bony collar. Okay. Then we get that blood supply that was, so now we're moving on to step 3. So then we get that blood supply that was coming close to the surface, starts to penetrate into the shaft of the bone. And into the shaft of the bone, this is what we call the primary ossification center. Primary meaning first. It's the first place we're actually going to get some true ossification. So when that blood vessel penetrates into this diaphysis, it's bringing with it fibroblasts, which differentiate into osteoblasts. Right? Again, wherever we have osteoblasts, we're going to be able to lay down bone. So now we're actually building bone in this middle area, this primary ossification center. And then step 4, we have what's called remodeling. Okay, so remodeling is now osteoclasts come in and they're following behind the osteoblasts. And as the blasts are building long ways and out ways, the osteoclasts are coming in and hollowing out the center of the bone, right? So now we're seeing our marrow cavity is enlarging and our compact bone is growing, right? Compact bone here, right? So those are first 4 steps. So our diaphysis is pretty well on its way to being like a mature bone, but our epiphyses are still cartilage. So what's next? Okay. So what's next is we get the similar type of process. So we get these little bony collars coming around the outside edge. We get penetration of blood supply into the epiphyses. And now we have what are called secondary ossification centers in our epiphyses, right? So now we have bones starting to form here, and we have bones starting to form here. These are secondary ossification centers. So then they're going to start radiating bone out this way and radiating bone out this way, and this is going to be primarily spongy bone because it's the epiphyses, right? Compact bone is going to be made here in the shaft. Spongy bone is going to be made here in the epiphyses. So it's going to continue to grow, right? Get bigger, bigger, and bigger. Bigger and bigger. So then what we end up with is probably about by the time the baby's born, you're going to have mostly the diaphysis is ossified, most of the epiphysis is ossified, but we have some leftover cartilage. So the cartilage on the surface of the epiphyses, this will eventually become your articular cartilage, the cartilage that stays to become part of your synovial joint, which we're going to talk about in chapter 9. And then we have a little layer of cartilage in between the epiphysis and the diaphysis. This is what we call our epiphyseal cartilage, or sometimes called the epiphyseal plate. Right? The plate of cartilage still there. And then as you grow, you can grow, sometimes these are called growth plates. Right? So as you grow, your bone can increase in length because you still have that epiphyseal plate. The cartilage is still there. Once you are mature, the epiphyseal plate closes, there's no more cartilage, and all you have left is a remnant of that growth plate, which is now called the epiphyseal line. So that's the closure of the bones. I believe in chapters 7 and 8, probably 8, because that's where your appendicular bones are. There's a whole table at the end of the chapter that gives you age ranges on when those epiphyseal lines close, like 10 years old, 12 years old, 18 years old. So that kind of gives you an idea as to the age of which the diaphysis finally meets the epiphysis, and we close off that cartilage plate, and now we just have the epiphyseal line. And that would be your mature bone, which if you're over 25, that's all of our skeleton would be that way. Okay. So here's some micrograph pictures, which I always like to show in class. So down here, this is the growth. That's not a good color. Let's see. Purple. I don't know if that's any better. So the diaphysis is growing this way towards the end of the cell, as far as the end of the bone, sorry. The epiphysis is all growing out this way. Okay, so it's going, that's a secondary ossification center. And then what we have, all of this is hyaline cartilage. It should kind of look like hyaline cartilage to you. And then here is our growth plate. So this is all cartilage. And so as long as that cartilage can be maintained, the bone can continue to grow in length. And once that closes up and these two bony parts meet each other, then that's when we're going to get the epiphyseal line. And here's just another micrograph picture of the same thing. So here is our spongy bone of our epiphysis. Here's our compact bones. The bone is growing in length this way, while the epiphysis is growing out this way. And then here is our growth plate. Okay, I think I have one more. Yes, so super duper close up of that epiphyseal plate. So all of this is cartilage. Here's the marrow, right? So here's our spongy bone. So this is the actual bone. Those are the trabeculae. And then the marrow is the hollow spaces in between. So this bone is growing this way, this bone is growing this way, and when those two places meet, we lose our epiphyseal plate and the bone can no longer grow in length. So here is a diagram, a picture of a fetus developing, and you can kind of see the diaphysis. The primary ossification center is completely, looks ossified, but where we would have the ends of the bones, they're not quite there yet. So we're kind of somewhere towards the middle of this whole endochondral ossification is the diaphysis has ossified, but those secondary ossification centers haven't been formed yet. So here's the femur, but there's no end of the femur. Here's the tibia, but there's no end of the tibia. So for this particular fetus, there still needs to be more endochondral ossification taking place. And you can see these big gaps in between the finger bones, because those are the epiphyses that haven't yet to be made. Okay, so then here are some x-rays showing the presence of those epiphyseal plates and then what it looks like once they close into epiphyseal line. So here we can see kind of the dark lines that show the cartilage, because cartilage isn't as dense as bone, so it shows up black, if you will, in an x-ray. And then we can see very subtle these lines in a mature bone. So this is your femurs on the top. So the femur is up here and the tibia is down here. And then we can also see the same thing up here in the upper picture. So for the thumb, we can see a good epiphyseal plate and then a corresponding epiphyseal line. Here's your radius, okay, epiphyseal plate, epiphyseal line. So a lot of times in what's called forensic anthropology, researchers and professionals use this information. If they come across a skeleton, they can take a look at the epiphyseal plates and epiphyseal lines and determine the age of the person when they died. Because if all they have is epiphyseal lines, then it was a mature individual. If they had epiphyseal plates, it was some kind of child, some age of child. If you want a good book on forensic anthropology, computer's kind of, there we go. It's called Dead Men Do Tell Tales. So if you want a good book to read over the Christmas break and you're super interested in the skeleton and it's kind of like a forensics, if you like forensic kind of science. So there's that saying, you know, dead men don't tell tales. Well, according to this forensic anthropologist, Dead Men Do Tell Tales. Very good book, lots of case studies, just amazing the kind of information that they can gather from skeletons. All different full skeletons to just little tidbits of bone that they're able to solve cases with. It's very fascinating. All right, so that is our endochondral ossification. I think, yes. So now we're moving on. Oh my, there we go. Intramembranous ossification. So this doesn't have, it's not quite as complex of steps. I know it shows 5 steps, but they're pretty continuous with each other. Intramembranous ossification is primarily for flat bones, right? So here's your skull, flat bones of the skull, your mandible, parts of your scapula. These are all formed via intramembranous ossification, where your long bones are, like your appendage bones, are formed primarily by endochondral ossification. So as the name implies, membranous, it starts off within a membrane. And what kind of membrane is, it's like a dense irregular connective tissue. Sometimes they're called dermal bones because the tissue that it starts off with is very similar to dermis of our mature skin. So remember mesenchymal cells. Mesenchymal cells were those connective tissue stem cells. So they kind of cluster together within this dermis-like tissue, and then they start differentiating into osteoblasts. Okay? Wherever we have osteoblasts, we can start laying down bone. So these mesenchymal cells cluster together, they differentiate, now they're into clusters of osteoblasts. The osteoblasts start laying down bone, and then you start growing these spicules of bone. Remember, the inside of flat bones are the diploid, the spongy bone. So that's basically what we're trying to make, are all these little branches of trabeculae. And that's exactly what happens. And so branches kind of get formed. Sometimes the blood vessels get trapped within the bone matrix, and then it just kind of keeps osteoblasting and osteoclasting, building and breaking and creating this dynamic until eventually you're going to get some nice spicules of bone with blood vessels or bone marrow trapped inside. And that's how you make the diploid of your flat bone. So this is kind of showing both your endochondral and intramembranous ossification from I think about 10 weeks of development to about 16 weeks. This might be a month and a half difference between these two fetuses. So here we can see kind of a review of our endochondral in the leg bone. So we just are seeing the diaphyses. We don't see any epiphyses. And then we can see this change of the femur here to how much is developed in just, like a month and a half. And same with the arms, you can take a closer look at all of those appendage bones. In the skull right here, you don't really see much. Intramembranous ossification produces the roofing bones of the skull, but in this 10-week fetus, you hardly see anything. It's like see-through, it's almost like a jelly. The brain is just kind of these clusters of neurons. It hasn't really formed. But take a look at how much progress is made of these flat bones. And there's the mandible right here. Red on red didn't really show up very well, does it? So those are the flat bones that start off as these dermal tissues, just kind of connective tissues. So it's a pretty amazing process, the process of your skeleton forming from either cartilage for your long bones or a dermal connective tissue for your flat bones. Pretty amazing. Here's a micrograph of that intramembranous ossification. So here we can see these are all the little spicules of bone, right? Kind of random shapes. And then inside, that's going to be the bone marrow. Here is your spicule of bone, super close up. Osteoblasts laying down, so the bone will be growing out in these directions as those osteoblasts are laying down their bone. Okay. And then lastly, I'll put this link because you can't click on this clearly, obviously, in this video. You can, if you wanted to type that in, but I'll make a link. There is a YouTube video of this disease. I can't think of the disease. Something progressiva, ossificans progressiva, or something along those lines. Basically, it's a disease where the individual's tissues, so like skin and muscle, turn into bone after some kind of injury. It's a really devastating disease. So here is this patient alive on the left-hand side, and then after he died, he donated his body for research. And so here we can actually see what was going on with his skeletal system underneath his skin. And we can see these huge plates of bone just kind of growing over the joints. And what happens is it kind of locks these people into an immovable skeleton, and eventually they'll die. But the video, if you wanted to watch that YouTube video, I usually show it in class, kind of illustrates the problems that are happening. So normally, normal skeleton formation is we're converting cartilage or connective tissues into bone, which is normal ossification. In this particular disease, it's called FOP. I can't remember what the F stands for. I can't even remember. Other types of tissue like muscle, after an injury turns into bone. And so it is abnormal ossification process. There are some other ones that aren't quite as severe. So these are called ectopic or heterotopic bones, where you get calcification and ossification of bones in locations that are odd or incorrect. Could be after an injury, could just be a weird calcification of your tissues. Okay, so that wraps up this video. I will see you next time. Bye.